Now, let's start the fundamentals of HPLC. This course is mainly focusing on the concepts and hardware of HPLC. This section describes the devices that make up HPLC. A HPLC system consists of various components, including a degasser, a pump, a sample injector, a column oven, a detector, and a workstation. Each component plays an important role. In this figure, the mobile phase flows in the direction of the arrows and everything is connected by single flow path. This diagram is called flow diagram. A mobile phase passes through a degasser before entering the pump. The degasser removes dissolved air from the mobile phase. The pump delivers mobile phase at specified flow rate. It prevents the formation of air bubbles caused by dissolved air, which can cause the pump to malfunction, resulting in fluctuation in the detector baseline. Then, the sample injector introduces a standard solution or a sample solution into the mobile phase. There are two types of injector: manual injector and auto-sampler. The auto-sampler is able to automatically introduce a specified amount of the standard or sample solution into a HPLC column. The column is placed in a column oven, keeping the temperature constant, and separates a target compound from coexisting compounds. A detector is needed to detect the separated compound by the HPLC column. A workstation processes the signal from the detector to obtain a chromatogram. Currently, a computer data system that performs both HPLC control and data processing is widely used. This is an overview of a typical configuration of HPLC system called Nexera series. The design of HPLC system is quite compact, as you can see. From this slide, each HPLC component will be introduced one by one. First one is solvent delivery pump. A stability of solvent delivery affects the precision of the retention time of compound and the calculation of peak area. A typical LC pump is based on the reciprocating piston design shown on the right side of the figure. The basic elements of the pump are a cylindrical pump chamber that holds the piston, a motor that operates a driving cam, a plunger seal, and a pair of check valves. When the plunger is pulled to the left in the figure, the inside of the chamber is depressurized. Then, the inside of inlet check valve is depressurized. Then a ball inside the check valve floats, making a gap between the ball and the sheet. This action allows mobile phase to enter the pump chamber. Conversely, when the plunger moves to the right due to the action of the cam connected to the motor, the chamber is pressurized and the inlet check valve closes. Then, the outlet check valve opens and the mobile phase in the chamber flows toward the column. The pump operates these processes continuously to deliver the mobile phase. Old type pumps have a single plunger with one pump head. However, the double plunger type pump has become a standard because it provides more stable flow than that of the single plunger type. In addition, the plunger size has been reduced for stable solvent delivery, and the LC40D shown on this slide has 10 microliter of plunger stroke. There are two types of elution: isocratic elution and gradient elution. Isocratic elution means that the mobile phase composition is constant over during analysis time. On the other hand, gradient elution means that the composition of the mobile phase is changing during analysis. When some compounds that have different retention properties are contained in a sample solution, it is difficult to separate these peaks with isocratic elution mode. In this case, the gradient elution method is highly recommended because the composition of mobile phase can be changed corresponding to the retention of target compounds. As a result, the analysis time could be reduced and the quality of the separation could be improved as well. Detailed explanation for isocratic and gradient elution is in the next page. A gradient elution is commonly used in reversed phase mode and ion exchange mode. This slide shows a comparison of the gradient elution and the isocratic elution in reverse phase mode. Two chromatograms on the left side are obtained by isocratic elution mode with 40% of methanol and 80% of methanol and water respectively. In the case of 40% methanol, 4 compounds were eluted from the column at appropriate retention time, but 2 others were held in the column for a long time due to the strong hydrophobicity. The peaks of these 2 compounds are broader than the earlier 4 compounds because they stayed and spread in the column. Then, in the case of 80% methanol, 4 compounds were eluted rapidly and the peak resolution was too low, but 2 others that have strong retention were eluted at an appropriate time and the peak shapes were much better than those from the case of lower methanol concentration. On the other hand, the 6 compounds were separated appropriately in shorter analysis time using the gradient elution mode. The composition of mobile phases was linearly changed from low to high ratio of methanol as you can see in the chromatogram on the right side. There are 2 types of gradient elution mode, high pressure gradient mode and low pressure gradient mode. High pressure gradient mode is composed of 2 or 3 pumps. 2 or 3 solvents are delivered by the individual 2 or 3 pumps. The mobile phase composition is controlled by the flow rate of respective pumps. A low-pressure gradient mode consists of 1 pump with a solenoid switching valve that enables the delivery of up to 4 different solvents simultaneously. Next one is sample injection devices. Sample injection devices are classified into 2 types, manual injector and autosampler. A manual injector is a device that introduces a sample into a column with a syringe manually. It is easy to handle the simple mechanism, but the accuracy of sample injection depends on the operator's skill. Then, the injection volumes and retention time tend to be less precise than those of autosampler. The autosampler is a device that affords automatic sample measurement, sample injection, and cleaning of needle. Although it is mechanically complex and maintenance is required, it is an important and essential device for consecutive analysis of large numbers of samples. This slide shows the structure of manual injector. The manual injector consists of a stator, a rotor, and a handle. When the handle is placed at the load position, the sample loop is disconnected from the mobile phase flow path and connected to the needle port shown as the upper figure. A sample measured by a syringe is introduced via this needle port. When the handle is placed at the inject position, the sample loop is disconnected from the injection port and connected to the mobile phase flow path leading to HPLC column. The injection operation is performed by inserting the needle of the syringe into the injection port, changing the handle position to load position, and pushing the plunger to introduce the sample into the sample loop. When the handle position is switched to inject position again, the sample in the sample loop is pushed with the mobile phase and introduced into the column. Normally, the timing of switching the handle position from the load to the inject is used as a starting point for the analysis and retention time measurement. In the case of injection using an autosampler, the operator has to prepare a sample in a vial and place it into the sample rack. Then, the autosampler executes injection following the method in which necessary settings are specified, such as injection volume created with workstation. The actual operating mechanism is a loop injection method, which performs the same operation as the manual injector described in the previous slide, or the direct injection method that injects the total amount of the sample taken from the vial by the sampling needle through the injection port. For both methods, the analysis starts at the operation timing of the valve switching. As expressed name of autosampler, our latest autosampler equips functions such as automatic creation of a calibration curve by diluting the sample with a specified solvent at specified dilution rates, and automatic derivatization by mixing the sample with a reagent followed by a certain standing time before injection. The function of a column oven is to keep the temperature of the columns constant. As described in the previous chapter, The separation of compounds using chemical interaction between the stationary phase and the target compounds is quite sensitive against temperature change. Also, even in analysis without using chemical interactions such as SEC, the rates of mass transfer and molecular diffusion are affected by temperature. Moreover, the peak shape and retention time are not stable without the temperature control. There are two types of column oven: block heater type and air circulation type. For block heater type, the column is heated on a metal block designed for a couple of assigned column sizes. Since the accuracy of temperature control would be worse if there is a space between the column and the block, installable column size and numbers are limited. For air circulation type, columns can be installed anywhere in the oven. Therefore, the size and number of columns that can be contained are more flexible than those of the block heater type. On the other hand, the size of oven could be large because of the space for circulation system. This air circulation type can be equipped with a mixer and a manual injector using the empty space of the chamber. It contributes to the stability of analysis in addition to the temperature control. That is all for this part. Thank you for your attention.
Excellence in science. Shimadzu.